Good morning. Happy Tuesday. Happy last week of the semester. Last full week of regular classes. Um, it's been a journey, but I'm glad we took it, and we're almost at the end. I think everything's gonna be okay. So for our last week, good morning, Monica. We're going to be looking at proteins, and we may look at the DNA RNA stuff today too, because depending on how long everything takes, and then we can use Thursday just for exam review. Morning, Daisy. So again, reminders for wrapping up the semester. Morning, Alyssa. The final exam will be next week. Morning, Maria. I've been trying to get the other two exams available for you to see the answers. Can anybody see the answers for exam 2? I can't figure out how to get that opened up. I think what I can do, I'll try today, is make a printout of it, like a PDF that has the answers, and then you should be able to see it that way. So it's not as ideal as being able to actually work through it, but I've changed all the options I can. I can't see how to do that. So I'll work on that some more today. At some point in in this week, you should be, let's see if it's up yet. You should be getting enrolled in a new Blackboard shell. So the way the final exam is going to work, because it's standardized across all the sections, it's not going to live inside our Blackboard. It has its own Blackboard shell, and it looks like it is not active yet. So at some point this week, there's going to be a new Blackboard shell that is only for the final exam, and you'll go in there, and it has honor lock and all the usual stuff. And that's how you'll do the exam. And I don't know the exact times yet, but I believe the exam will be open something like Monday through Wednesday next week, and you can pick your time to complete it. And I believe you have about 2 hours to complete it. All right. So let's talk about proteins. So this last bit, similar to what we did last week, is going to focus on looking at the structural and identifying features of these biological molecules and some things about the structure and function, but mostly descriptive, so rather than like problem-based. So let's take a look at some proteins. Now today I'm going to be looking through and using the book for reference, because again, it is pretty descriptive. So there's going to be some structure drawing, but less than we had done before. All right. So what are proteins? Proteins are a class of molecule that do a whole bunch of different things in the body, and you can have many different roles for proteins in living things. So there's a table here with some of the different types of protein types for some examples. So there's enzymes, digestive enzymes that break down macronutrients and other things like that. There's transport, so proteins that carry things around the body. Structural proteins that produce or are used for a structural or mechanical component to like hold things up, or like all kinds of different things. Hormones are molecules that coordinate different body systems activities. Some hormones are small molecules like the steroids that we looked at, and some are proteins like insulin. And then there are defense proteins, immunoglobulins that can protect from pathogens, contractile proteins, so proteins that move, and like muscles, which is what muscles are made out of, and then storage proteins, so things like egg storage or egg proteins that store nutrients for embryo development. So lots of different kinds of functions for proteins. depending on how they're put together. So there are globular proteins, so proteins that are kind of just like a blob, versus fibrous or more kind of long and skinny type proteins. And the structure of the protein, the shape, is really critical to how it works, to what it does. So we're going to look at protein structure or protein shape, kind of starting from the bottom, the smallest units, up to the large unit, the full protein. Another word you should know here, denaturation. Denaturation is when a protein kind of unfolds itself. So as we'll see when we talk about the structure, one important aspect of protein structure is how that protein actually folds and is arranged with hydrogen bonding and things like that, chemical interactions inside the protein, and you can often change that and make it do something different, and that's called denaturation. So one of the labs we were supposed to have been doing this week, if we would be in person, would be to take some milk and actually take the proteins in that milk and cause them to denature. And you may have done this either on purpose or by accident before. If you acidify milk, you can try it at home. Put some vinegar or some lemon juice in some milk and stir it around for a little bit. And eventually, what you'll see is the protein separating out and kind of congealing and forming chunks. This is how cheese is made. And that chunking is the proteins unfolding, and then the intermolecular forces kind of causing them to all tangle up and form these new clumps. So that's known as denaturation. You can also denature with heat. So when you, for instance, cook an egg, you're denaturing those proteins using heat. All right. So let's go down to the very basic, smallest part of the protein structure, which are the amino acids. We mentioned amino acids a little bit in the past when we were talking about functional groups. Well, let's take a look at amino acids again. Here's kind of a ball and stick model of what an amino acid might look like. It will have two basic carbons in the main part of the structure here. Let me make this a little bigger for those of you looking on smaller screens. All right. So there's this kind of main carbon in the middle, which we call the alpha carbon. It's called the alpha carbon because it is one away from the functional group carbon, the carboxylic acid carbon. So one away is alpha. So you've got a carboxylic acid here, carbon double bond to oxygen, and then OH. Then this alpha carbon, and then on the other side of the alpha carbon is a nitrogen. So that's the amine, that's the acid, and that's where that name comes from, amino acid. Now the alpha carbon is important because this part that's connected to the alpha carbon, which is known as the side chain, is what gives all proteins their, or gives amino acids their variation. So different side chains mean different amino acids. The rest of the structure stays the same for all amino acids. So let's take a look at some of those different side chains. Now, some of these names you may have heard before. Some of these amino acids: glycine, alanine, valine, leucine, methionine, isoleucine. If you take a class in um, biochem or something like that, maybe nutrition, you may have to learn actually all of these names, like which structures or which names, but we're not going to do that in this class. But you can see what gives them the different names, what makes them different from each other, are the side chains that are connected. So the simplest amino acid just has a hydrogen there, and that makes it and that's called glycine. Then you can have like a methyl group there, alanine. In the book here, these are grouped in the way that they are commonly grouped, which is by their polarity and how they interact. So you have your nonpolar aliphatic R group. So nonpolar, we know nonpolar means. Aliphatic means single bonded carbons. So things like CH3s and CH2s and CHs, carbon chains basically. And methionine is put in there too, because yes, there's a sulfur in between the carbons, but it's still mostly carbon nonpolar type stuff. Then you can contrast that with the polar R groups. So these are going to be things with OHs, SHs, NHs, those sorts of molecules. There's one kind of strange amino acid in this group, which is proline. And I say it's strange because it's the only one that deviates a little bit from that basic structure, and that the side chain is actually also connected to the amine. So you can see that forms a ring here. Um, Whereas all the other ones, they just have this group hanging off the alpha carbon. This one, the group coming off the alpha carbon, also connects over to the nitrogen. And that has some really important structural significance. Proline is a really common amino acid because it causes the chain to kind of turn because of that linkage, that five-membered ring linkage. All right, these other categories on the right, there are some side chains that have positive charges on the R group, so nitrogen-based um, side chains with positive charges, some negatively charged carboxylate-based ones, aspartate and glutamate, and then what we call aromatic R groups. Aromatic, as you can see here, are these ring structures with alternating double bonds. So that's sometimes called a benzene ring, and so these types of amino acids, phenylalanine, tyrosine, and tryptophan, are aromatic. Now, another interesting thing about these, you can see that they're listed as non-polar, even though tyrosine has an OH and tryptophan has an NH. So in this case, again, it gets a little bit more subtle than just saying if you have an OH, it's polar, or if you have an NH, it's polar. It's really more of a balance of the whole molecule. This is going to have a polar part here, but because it has this really big aromatic ring, all this carbon and hydrogen there, that kind of outweighs it. And when we think about this as a whole molecule, it becomes more of a non-polar side chain. So that's the classification. You don't have to be able to memorize these names or know where they go, but you should be able to recognize the functional groups, and if you're given a structure, be able to classify it as polar or nonpolar based on the side chain. All right, so those are different amino acids. Another thing to look at here, if we compare what's in this table to the structure up here. So when we looked at this structure, we see NH2 and then CO2H, which are the traditional kind of neutral ways that we write these. But because of the acidity of carboxylic acids, CO2H, in a neutral, like a water environment, a neutral pH environment, you're actually going to have a carboxylate. So you're going to have the conjugate base of the carboxylic acid where it's lost its hydrogen, and you're going to have the conjugate acid of the amine. Because remember, a carboxylic acid is quite acidic, and an amine is basic, like ammonia is a base. So the hydrogen is not going to stay on that acid in neutral conditions. It's actually going to end up transferred over to the amine. So the more correct way to express amino acids at a neutral pH is to have it a negative charge on the carboxylate and a positive charge on the amine, as you see in the table here. Now, at very low pH, acidic conditions, the carboxylate would also be protonated, would also have a hydrogen. And at very high pH and basic conditions, then the ammonium here would be deprotonated and you'd have NH2 and it would be neutral instead. All right, so that is the amino acids. Now, the amino acids also have, I don't think it's in this book even because it's not, you don't need to know them, but the amino acids also have abbreviations. Abbreviations that are both, I think we'll get into this in the next part actually, that are both 3-letter abbreviations and 1-letter abbreviations. Again, these are not things you need to memorize, but they may come up in your future learning, and you can recognize that they are in fact that they do exist. So, yeah, here's an example. So, alanine can be designated as ALA or just A, and you can look up those if you need them for some reason. You won't for our class. All right, so let's take these amino acids then and show how we build up what we call the primary structure of a protein. I'm going to switch back to drawing. The primary structure of the protein is the sequence of amino acids. Right? So we're going to look at four different levels of protein structure: primary, secondary, tertiary, and quaternary. And that's just kind of as we get bigger and bigger, kind of like zooming out. So the primary structure is the sequence of amino acids. So the how the amino acids are connected to each other. And as we've learned before, they're connected by peptide bonds, which are the result of a dehydration or condensation reaction between the carboxylic acid of one amino acid and the amine of the other. So we're going to write them in their neutral form again, just because it becomes easier to see. So let's pick a couple. Let's look at let's say alanine. I'm going to draw the whole thing out here. So alanine has a CH3 as its side chain. And let's connect that with, let's pick a big one here, or a bigger one just for fun. How about tyrosine? So if we want to condense this with tyrosine, tyrosine has a CH2 and then that aromatic ring and the OH. All right. So we do that same reaction that should be very familiar to you at this point, where we take two hydrogens and an oxygen, get rid of them, and then connect the remaining carbon to the nitrogen. So if we do that, we now have these two amino acids connected via an amide or peptide bond. All right, so something like this. And we might call this alanynyl tyrosine, I think is how you would describe that. So this would be what we call a dipeptide made of two amino acids. Let's highlight this important peptide bond here. So we call that an amide or peptide bond functional group. That's how those things are linked. And because the two ends over here on the left and over here on the right have the same functional groups as any one of the amino acids, then we can link more and more. We can keep linking off from the left, and we can keep linking off from the right. See that? All right. So the as you can see, we could keep building that on and make a big chain, and that's the protein sequence. What is the order of amino acids connected by these peptide bonds? Functionally, they're all the same. Where they differ is the side chains, and that's known as the primary structure of the protein. All right. Any questions about how those link up? So good question. When do they stop linking? I mean, it just depends on the protein. So there needs to be some way for whatever is directing the protein to build in that particular way, which is generally the DNA and RNA, how that works. Something else in the body has to tell them. Some other molecule has to say, okay, put this thing together, and then stop. And that's generally the job of the information in the nucleic acids, is to kind of dictate how that protein is put together, and that's to make that primary structure. But you're right. If we just sort of mix these things together with enough energy or catalysts or whatever, they would just keep linking up in a random way until it was all whatever the material was all used up. So there has to be some sort of mechanism for putting them in the correct order, and that's what the nucleic acids do. All right. So this is oh, sorry, I'm gonna change my view again. So this is the picture from the book again. Here's an example of kind of zooming in on that primary structure, where we have a whole bunch of amino acids linked together in a particular order. The primary structure, in this case, this is bovine serum insulin. You'll notice that the cysteines, CYS here, are linked, actually link two different chains together through something called a disulfide bond. And so this actually gets into the next part of the structure, which is the secondary structure. So if you, I'm not going to do it right now, but if you were to jump back to the actual protein structure, the amino acid structures, you'll notice that some of them have SHs, like cysteine. And so SHs can actually link together to make these SS bonds. And that's one way that chains can link. And sometimes those links are between different chains, like here and here. And sometimes those links are in the same chain. You'll notice this is really stretched out, but in reality, that would cause that chain to bend back in a particular way. This is, for example, how you can mess with the curliness or lack of curliness of hair. The curliness in hair is often caused by these disulfide bonds. So if you break those bonds, like chemically, like with relaxers, you can cause the hair to stretch out and straighten up. You can also introduce more of those disulfide bonds, which is sometimes called a perm, where you're making those bonds, and that causes the hair to link up and curl in that way. So based on what that sequence is, that protein is going to shape itself in a particular way. So take a look down here at these pictures, and we're really just going to go from the book here because these pictures get really too complicated to draw. So we'll use their figures. There are also some neat videos. I've linked some in Blackboard, and you can find other ones on YouTube, kind of showing three-dimensional examples of what these things look like, because they are quite big, and it's something that's difficult to draw. So what happens here in the secondary protein structure now, we're no longer talking about the chemical bonds of amino acids together. So now that we have this chain, the chain doesn't just flop there straight. It actually twists around and forms a new type of a structure. Oh, the link from Blackboard cannot be found? Okay, yeah, I will, let's see. Let's take a look at that. Maybe I typed in the link wrong or something. I think it's just another chapter of the book. You can just navigate through the book itself, like I've been doing here with these buttons on the bottom. But yeah, there's probably just a typo in the link or something. I'll try to fix it. All right, so when you've made these chains, remember these chains are full of NH bonds, of oxygens, of sometimes with these sulfur bonds, other things. So they can actually arrange themselves in a way that maximizes the intermolecular forces. Although in this case, it's an intramolecular force because it's in the same chain. But the two main components of secondary protein structure are the alpha helix and the beta sheet. And here's how that works. In the alpha helix, what happens is if there are NH bonds that are a certain number of amino acids away from a CO, and there's generally a proline in there that is causing it to turn a little bit also. Remember, the proline is that one that links together. So if those are in the right position, then this kind of twists back on itself so that the H and the O can line up. Because remember, H is connected to N's, can hydrogen bond with oxygens or other nitrogens. So you get this hydrogen bonding interaction that kind of locks this thing into place, and that continues throughout the chain for a particular for various different sequences. And if you keep doing that over and over again, you have what's called an alpha helix, which is this part of this protein structure. And it's called secondary because, remember, the primary structure is the actual order of amino acids, so like which amino acids are connected to which. And the secondary structure shows how that chain kind of orients itself. And the two main components of that are the alpha helix and the beta pleated sheet. So there are, like with the alpha helix, one way for those hydrogen bonds to link up is to twist around and link up that way. Another way is for the sheets to be mostly flat. They're not exactly flat. They're wavy because of the bond angles, but they can also arrange with another part of the chain and form these kind of flat sheets, and those are called beta sheets. And when they're drawn, they're expressed in this way, where it's like a flat ribbon with an arrow at the end, and the arrow at the end kind of orients it into which direction it is. So go ahead, Jennifer. Back to the order of amino acids. Go like this. Yeah. Right. Yeah, it's determined by RNA or synthetic. Like, it's determined by whatever is determining it. You could go in the lab and you can build your own chain of amino acids however you want. For example, if we're looking at like the COVID vaccines that are coming out, those were developed. The new ones are using like an modified RNA that goes in and has your cells make these COVID related chains. But you could also do this. Another vaccine technology is to actually go in the lab and make a specific chain that mimics a piece on the virus and then put that into the body and have the body react to it. Yeah, in the cells, again, depending on the type of, so that's like a specific example, but these things are just molecules that can be made in various ways. In fact, now there are machines that you can, like essentially printers, that you can type in the protein sequence you want, and the machine will make it for you. You can actually do this online. You can order your own protein sequences from companies that will put them together in whatever order you want to do that. All right, so the secondary structure. So these are two parts of the structure. Now notice the way this is drawn here. You can see this is all still just one molecule. So one super long chain, super long chain, right? Hundreds, thousands of amino acids, whatever, is arranging itself into these different other structures based on how amino acids are placed in proximity to each other. So these kind of wind up or arrange themselves in these sheets in a particular way, but we're still talking about one whole chain. And that's what we call the secondary structure. All right, so then that moves us on to the tertiary structure. So tertiary means like third. So we've got the first level, the primary, which is the sequence of amino acids. The secondary, the alpha helices and the beta sheets. So these larger pieces that get arranged. Then the tertiary structure is how the whole thing, these beta sheets, alpha helixes, other stuff, the sometimes called the active site, or if there's a metal inside the protein that kind of does its thing that it's supposed to do, how all of that now arranges itself into one big giant piece. So that might be something like this, and there are several or just a few things that determine this. One is ionic bonds. So if you have a negatively charged side chain, a positively charged side chain and a negatively charged side chain, those are going to attract each other and cause that whole chain to fold up. Now, to show this more clearly, this is just shown as like a noodle or something, but actually this noodly part, that's your secondary structure. That would be the alpha helixes and the beta sheets. And so the negative and positive residues are kind of forced to come together in that way to connect as an ionic bond, and that folds that protein in that particular way. Another way that folds is called hydrophobic interactions. So when you have non-polar side chains, remember much of the body, where these proteins are going to be is in a water thing. I guess not always, but primarily it's going to be in water. And the chain itself is quite polar because of all the hydrogen bonding and everything. So the nonpolar parts are a little bit out of place. And so they find each other and they want to attract each other. So nonpolar things like these valine carbon and hydrogens, or aromatic side chains with the benzene rings, those are going to want to find each other and attract each other. So that's another thing that can cause it to fold in a particular way. The disulfide linkages that we talked about before are another way that like cysteine residues can kind of find each other and form this sulfur-sulfur bond. And then the final one is there can be more hydrogen bonding. So just like the hydrogen bonding within the helixes and the sheets in the secondary structure, there can be hydrogen bonding that pulls this whole thing into a tertiary structure. So all of those things are happening in the protein, causing the protein to fold in a particular way. So it makes this new kind of structure based on how all of those things interact. And that's really what dictates the, in many cases, the function. So all like the sequence, the amino acid sequence and all of that is important. But for many proteins, it's really that overall final structure, or the overall tertiary structure, that determines its function. And so if you mess with that structure, if something else messes with that structure, that's when things start to go wrong. So that's so things like many types of diseases, especially like genetic diseases, damage, like when you have things like UV ionizing radiation damages DNA, and then it doesn't code the protein quite right, and then all of a sudden it can't fold properly, and now it can't do what it's supposed to do, and then weird stuff starts happening that can lead to uncontrolled cell growth, which is what we call cancer, and things like that. So when the proteins can't fold properly, there's big problems. When they're in environments where they don't fold properly, then they start to potentially do other things or cause other problems. And because there's so much involved in a protein, it's so big that how a protein folds is actually a really complicated problem that's just now starting to be better understood. Even with really powerful computers, simulating protein folding is a complicated problem. And a couple neat, if you're interested in this, a couple neat resources. There's one thing called Folding at Home, which is basically an app that you run on your computer. And when you're not using your computer, it uses some of the computing power in the background to help scientists. I guess it doesn't really show much here. To help them figure out this protein folding problem, because you need so much computing power to solve these problems with all these interactions and trying to optimize all these interactions, so much power that one way to get all that power is actually to distribute it. So to have a bunch of people's computers working at the same time on these problems. So if you start this up, you're actually using your computer to help scientists figure out protein folding problems, which helps us fight disease. Another neat one that is a little bit more active is called fold.it or foldit. That is a program that you can download on your computer, and it's kind of a game where you fold proteins with your mouse and try to find the best interactions. And the reason for this is it turns out that this is such a difficult problem. It's so There's so many degrees of freedom. There's so many different ways you can twist and pull and arrange these things that just throwing it at a computer and saying computer do it doesn't doesn't lead to the best results. And it actually works better if a human with all of our processing capacity in our brain, if we notice these types of interactions, hydrophobic interactions, hydrophilic interactions, whatever, and try to fold these things ourselves first, and then say, okay, here's a good candidate, and then let the computer just optimize it a little bit. And there's been a lot of neat advances, including with COVID, with this particular program. So people download this, they try to get high scores, so it's kind of like a game, and by getting a high score, you're actually reporting the best ways to fold these proteins. And then scientists can take that information, try to actually do those things in the lab, and use those results to come up with some real important results. So if you're looking for something to do after the semester's done and you want to solve some protein folding puzzles to help out science and to help out with some vaccines and things like that, this is a neat one to play with. It doesn't require any specialized scientific knowledge. It's meant for just like kids can play it. It's just pattern recognition and like twisting things around, but you are actually helping by playing that. So those are some ways to look at protein folding, to look at those tertiary structures of proteins. All right. And that brings us to the quaternary structure, which is generally a little bit less important, but quaternary structure is when multiple protein chains, so multiple tertiary structures, pack together to make one. So there are many proteins whose quaternary structures are their kind of functional form, both in the solid state and not. But up to this point, up to the tertiary structure, if you see here, this has all been one chain. So we started with our one chain. It arranged itself into these helix and sheet things. Then it arranges itself into the tertiary structure where it folds in a particular way. And now we're actually changing to multiple of these. So multiple pieces of the same thing could pack together in a particular way. And that's that quaternary structure. So denaturation is, as we said at the beginning, when you undo all of this. So when you unfold things back to the primary or secondary structure using things like acid and heat, which so acid does it because it changes the protonation state, so it changes whether things have hydrogens or not, and that will break some of those interactions. It will also mess with disulfide bonds and those kinds of things as well. And then heat gives it enough energy to break a lot of those intermolecular interactions, like the hydrophobic interactions and the hydrogen bonds, so that the heat causes those things to break apart. And then once they're broken apart, it's often difficult or impossible for the protein to refold in exactly the same way. And so that's why they tend to get tangled and clumpy and whatever once that denaturation happens. Now, of course, there's lots of different kinds of enzymes. So, for example, here, stomach's digestive enzymes need to keep their function, their shape, and their activity under really low pH because our stomach's pH is really, really low. So we can denature all these other proteins that get into our stomach, but the digestive enzymes have to keep their shape. So those are proteins that fold. Their folding is optimized for that low pH. So if you perhaps raise the pH, you could mess with the digestive enzymes in that way. There's some animations you can look at here. But that's the basics of protein structure. So if you look up just kind of in a general search, you'll see some some various ways that protein structure is expressed in just in examples. And it may look very, let's take a look at some random spots here. It may look very strange, very like chaotic. I always thought that when I saw a structure like this, I just had no idea what was going on. But now I know a little bit more, and I know I can recognize these alpha helix pieces, so I can recognize these bits of secondary structure here. I don't see any beta sheets in this example, but you can see how those helixes are and the other parts are kind of folded in particular ways. So the fact that like this one is here and this one is here and this one is going this way and this one is going in this way, it's not just random. That's folded in the particular way that's dictated by the side chain residues and how those things are connected. And then we have in this case an active site. So this is something enzymatic maybe or like a heme protein or something like that, where you have some metal in the middle that is attracting. I think this is heme because it looks like it's connected to oxygen there. So that's an example. You can see lots of these other kinds of things here. Again, as an example. Right, so like various proteins. So here you see some of those beta sheets again, connected by some other stuff. A lot of these pictures are really pretty because not just to make them look pretty, but because it helps us understand and to notice, to be able to tell the differences in the different parts of a structure here. So you can see these kinds of things. And these are all examples of tertiary structures. If we get into the quaternary structures, I'm trying to see, so something like this maybe. Let's see what this picture looks like. Thanks. Well, it looks like I can't get to that exact picture, but what do we have here? Okay, no, I'm in the wrong kind of site. But anyway, so pretty pictures for proteins in their tertiary or quaternary structures. All right, so back to the objectives then. And as you're both learning new stuff and also reviewing old stuff, make sure you always keep an eye on these because this is ultimately what's going to be on the final. We should probably also note occasionally throughout the semester with quizzes, definitely with homework, sometimes with quizzes and sometimes with exams, we've gone a little bit beyond the specific objectives for each part. The final exam won't. So it'll be exactly on these objectives from the whole year. So we looked at some functional groups of amino acids, peptide bonds, reactions showing those condensations, and then we looked at the different structures and talked about denaturation and some proteins. We didn't really talk about substrate binding and inhibition of enzymes here. So enzymatic, there's not a whole lot. I mean, there's a lot to say about that, but for our purposes, what that means is, so we talked about enzymes being catalysts, lowering activation energies. And the way that that works is there's this active site in the middle of the protein. I mean, it doesn't have to be in the middle, but somewhere in the protein, there's some sort of a metal or something chemically active that can bind a different molecule and allow it to undergo a particular reaction. And that's known as substrate binding. So the substrate in that protein binds a particular molecule and then causes that molecule to react with another molecule and do a particular process. So some reaction that needs to happen in the body. So generally those enzymes are labeled with or are named with ASE at the end, so ACE. So a protease is an enzyme, a protein that can take another protein and cause it to break apart in a particular way. An amylase enzyme is an enzyme that can take amylose, a starch that we talked about last week, and break it apart into glucose. So things like that are enzymes that do a particular function in a body or in a cell or whatever. You can um, you can inhibit those, so you can make them not do that by designing other molecules that take those places. So if we want to stop, let's say, a bacterial or a viral enzyme from doing a thing, we can figure out what that protein is, look at its active site, and find or design a new molecule that will fit in there and inhibit it, stop it from doing its job. And that's one way that we fight illnesses, bacteria and viruses. For example, if we look at HIV, the virus that causes AIDS, that particular virus has a protein in it that I just forgot the name, but there's a protein in it that causes it to reproduce, and you can and some of the effective drugs for that protein inhibit that reproducing enzyme and stop that virus from reproducing in that way, or slow it way down. And that was one of the first drugs that was actually designed by looking at a protein structure. So when we look at those folding games and stuff that I told you about earlier, one reason that is so important is because if we understand how that active site works in that particular protein and how it's folded, then we can design molecules that fit really nicely in there and inhibit that protein. And then we can potentially stop like a virus from reproducing, for example. All right, question. Don't see quiz 9. Yeah, as usual, the quiz is going to come up later in the week, so Wednesday or Thursday, and it'll be due on Sunday night. Okay, so we will stop there for now. We'll talk a little bit about molecular genetics, DNA and RNA on Thursday night, and bring exam questions as well. We'll do some review sessions for the exams also. I'm thinking like one time over the weekend and then maybe on Monday or Tuesday. I have to get there's no mastering chem for this topic. It didn't seem helpful when I was going through it. There weren't really helpful questions. As long as you can kind of recognize the things that we talked about. If I find, maybe I'll put like an option. I'll see if I can find some questions and just put it up as an optional practice. So you have something to work through, some problems to work through for that. Um, but yeah, we'll plan on a review session sometime this weekend and then sometime early next week, depending on when the exam is scheduled for. I think it's supposed to be Monday through Wednesday, but I don't have confirmation on that yet. I think that's about it. Otherwise, if you have completed mastering chem assignments late, that's fine. You still get credit for them, but check the grade book in mastering chemistry first before Blackboard. And if there are assignments that are totally done, but you didn't get credit for them because they were late, let me know so I can change that due date, and you should still get full credit for late mastering chem. All right, I'll put some stuff up there as a practice or an optional assignment. All right, other questions or things you want to talk about before we go today? Yeah, I'll figure out a way to get exam 2 answers posted. Yep, you'll know the cumulative grade before the exam. It'll be that weighted total in Blackboard. I'll just get everything else updated in there. Also, if you want to meet with me in a less like on WebEx or something where there's a little bit more room for interaction, that's fine too. We can definitely schedule time to do that. It can be one-on-one, or like if a few people want to meet at the same time and discuss it. If like typing in questions is a little bit tough for you, or just this time doesn't work great for you, we can do that. All right. Well, I will talk to you all soon. Happy studying, and let me know if you need anything. Thanks, everybody.